Hello everyone, welcome to day 9 of genetics. Here we are going to discuss a very important topic. Actually, most of the MCQs, multiple choice questions, which are asked from biochemistry, from genetics, all is based on this what table only, that is lysosomal storage disorders. Fine? Yes. Even though we have told that it's the lysosomal storage disorders, the main problem or the main defect in all these disorders is there will be defect in degradation of a compound. As a result, the preceding compound will start accumulating and this is responsible for the clinical manifestations. Fine? Yes. So if you remember the enzyme defects which are mentioned in this table, definitely we can answer almost all the multiple choice questions which are asked from this genetics, because they will give a clinical case scenarios in detail, that is the in-depth detail of all these lysosomal storage disorders we will see in the subsequent videos. But now I have to focus myself only on this table. So as far as glycogenosis is concerned, that is glycogen storage disorders are concerned, the most important thing is Pompey's disease. Okay, the most important thing is called as Pompey's disease. Remember, the Pompey's disease is the only GSD which is an LSD. That means Pompey's disease is the only glycogen storage disorder which is actually a lysosomal storage disorder. Why? Because the enzyme defect, the enzyme defect is alpha 1,4-glucosidase, which is nothing else but a lysosomal glucosidase. So because of that, there will be glycogen accumulation. Don't worry, Pompey's disease, its clinical features, everything, histopathological finding, everything we will see in detail. Then we have sphingolipidosis. In sphingolipidosis, we have GM1 gangliosidosis and GM2 gangliosidosis. As far as GM1 gangliosidosis is concerned, the most important thing is beta-galactosidase. Beta-galactosidase is the enzyme deficient. In type 1 is infantile, type 2 is juvenile. Again we will discuss Tay-Sachs, everything in detail. Then GM2 gangliosidosis includes the three most important entities: Tay-Sachs disease, Sandhouse disease, and GM2 gangliosidosis AB variant. Yes. Out of that, Tay-Sachs and Sandhouse is most commonly asked. In Tay-Sachs disease, it is hexosaminidase A is defective, whereas both A and B are defective in case of Sandhouse disease. In both things, what GM2 gangliosidosis is accumulating. Fine. Whereas in GM1 gangliosidosis, it is beta-galactosidase. So galactose containing oligosaccharides will be accumulated. Then we have sulfatidosis, that is aryl sulfatase A deficiency, which will lead to metachromatic leukodystrophy. Metachromatic leukodystrophy is due to aryl sulfatase A. Yes. Then we have Krabbe's. Krabbe's disease is due to galactosyl ceramidase deficiency. So galactose cerebroside, is going to get accumulated. Then Fabry's, it is alpha galactosidase A, whereas beta galactosidase, it is GM1 gangliosidosis. Alpha galactosidase is Fabry's disease, so ceramide will accumulate. They will ask, in which of the following deficiencies ceramide will accumulate? Fabry's disease. So what is the next question in Fabry's disease? What is the enzyme defect? Alpha galactosidase A. Gaucher's is very commonly asked question. We have crumpled tissue paper appearance and all. It is glucocerebrosidase. So glucocerebroside will accumulating. Niemann-Pick disease type A and B, sphingomyelin is enzyme deficiency leading to sphingomyelin accumulation. And remember a very, very important point. Once again, I am repeating that point. Pompey's disease is the only glycogen storage disorder which is a lysosomal storage disorder. In GM1 gangliosidosis, beta-galactosidase is the enzyme deficient. In Tay-Sachs disease, hexosaminidase A is deficient. Sandhouse disease, hexosaminidase A and B is deficient. Then we have Krabbe's, it is galactosyl ceramidase. Fabry's, alpha galactosidase A. Gaucher's and Niemann-Pick disease we will discuss in detail again. Then next one is called mucopolysaccharidosis. We have mucopolysaccharidosis, that is mainly one, that is the Hurler syndrome and Hunter syndrome. Of course we have lot of other mucopolysaccharidosis also, but Hurler and Hunter are most commonly discussed. Hurler, it is iduronidase enzyme deficiency, whereas iduronate sulfatase is the enzyme which is deficient in Hunter syndrome. There will be accumulation of all these are disorders of mucopolysaccharides, glycosaminoglycans disorder. Then we have eye cell disease or inclusion cell disease. This is because of the defect in mannose 6-phosphate. There will be defect in lysosomal targeting. Then another most important thing is what we call as Woolman's disease. Woolman's disease, there will be cholesterol esters and triglycerides accumulation because the enzyme deficient is acid lipase. So what I have highlighted, these are the most commonly asked questions in all, whether it is the postgraduate entrance exams or in your MCQs, in your prof exam. These are the most commonly asked questions. You should know, okay, what is the type of inheritance that we will see in the what corresponding disorders we will see what is the type of inheritance. Next, what you have to remember is what is the enzyme deficient and what compound is going to get accumulated. So please go through this table again and again. I know it will be a little bit confusing, but once you revise it again and again, once you repeat it again, you can paste this table in your study in your room, so that you can revise it frequently, because this is you will forget very easily. So please go through this. Remember Pompey's, then Tay-Sachs, Sandhoff's, Niemann-Pick, Gaucher's. These are the most commonly asked. Niemann-Pick and Gaucher's are standard short note questions. Okay, we will discuss about all these things in detail in the subsequent videos. So I hope this session has been useful for you. Thank you.